really bad for the director. His leg caught in there. Anyway, I called the zoo director and everything about it. But is that the one that died from an injury?
I cannot claim nor deny. I'm not sure. Was it 5 years ago or so?
Yes.
They just contacted me and I'm like, you've got to be kidding me. They happen to be certainly. I'm sure. Well, I'm sorry to hear that you saw that. Well, okay. So what's really interesting about these infections is that when people come down, the symptoms, the initial symptoms are flu-like, and they're very confusing because people have very limited or mild pain or rash associated with the site of infection. And so, most infections, again, if you cut your skin, it's highly inflammatory. It's clear red and swollen where you're infected. These, you can barely even tell where you got infected. And so with these young women that are giving natural childbirth or overnight abortion pill, they come in complaining of flu-like symptoms. Because, again, there's no fever, no rash, and so they say, well, you just had a baby. Let's go home and see if you can shake it off and come back tomorrow. Well, if it's not caught immediately, we're talking hours to maybe a day. These patients go into, they develop local necrosis, multi-organ failure. They develop what's called a leukemoid reaction, and that was all my research. Their white cell count elevates to an extremely high level for reasons unknown. And then these patients go into shock and they die. It's very quick, and it's because of multiple exotoxins produced by the pathogen. And so here's a list of potential toxins that they can produce. I will say that most strains of Sordelia, if we were to go out here and dig them up, don't produce any toxins, and so they're not pathogenic. However, there's the few that produce kind of a hodgepodge, if you will. It's not they produce all or none, but usually they produce some of this list here. But the first two, the lethal toxin, the hemorrhagic toxin, those are the heavy hitters for this pathogen, and if they're producing these, that's what causes all the damage. And if you were to do a literature search today, you'll find that the LT and the HT take up most of the literature with this pathogen. We've done work with the neuraminidase, I'll show you some of that. But the role of these others, the hemolysin, et cetera, it's not clear, but it really hasn't been researched either, so we're not sure. So this is a young woman, his name was Dr. Jamie McGregor. He was a physician in Southern California that had the misfortune of working with one of these young women that had used the overnight abortion pill. And so he let me use a couple of these pictures. And so what happens, that lethal toxin and that hemorrhagic toxin, what they do is they cause rounding, cell rounding of your cells. And so cells of the vascular system that keep things closed, they start to ball up and round up and shrink. And so you start to drain, your capillaries drain fluid. And at the peak of this infection, these patients receive liters of fluid literally to try to give something for their heart to pump, and they just drain from the inside out. And so they start to look like the Michelin Man. And so that's what this poor woman was going through. There's a picture of her vagina. It was just, they can't stop the swelling. But again, notice how there's no real rash. It's not red or inflamed. It's just swollen, okay? Another fingerprint or hallmark feature for these infections. This is a slide of the tissue from this patient. You can see the purple rods there. That's the Sordelia organisms themselves. Another thing too, suppose are with these infections in humans, is it takes very few bugs to cause fatal infections, which is scary now because you have few bugs, and it doesn't, when you're sick, you don't give these clues or signs or symptoms to show that you're sick, and you die within a day or two. I mean, for aspiring young terrorists, I think this is a perfect, really a bioterrorist, organism, because it's confusing and it's highly lethal. And again, it only takes a few organisms to cause these infections. So this is that same review article we put out. Again, we were comparing all the vital statistics between the vital statistics between survivors and non-survivors. And as you can see, what was interesting to us was the fact that there was no difference in temperature. 37 degrees is a normal body temperature for humans. As you can see, survivors and non-survivors really didn't mount any sort of fever. In fact, the survivors developed a little more than the non-survivors. But the biggest difference was this white blood cell count. If you survived, you had a relatively mild white cell increase. But if you didn't survive, the normal count is around 2 to 3 to 5 times 5,000. And if you get a normal infection, it may go up to around 10,000 or so. People were getting around 75,000 white cells per microliter of whole blood, if you guys are familiar with a microliter, very small volume. And then some patients were as high as 200,000, so extremely high. Yes?
So that's like initially how you tell they had it, right?
Again, it's a fingerprint, absolutely. And so I think, well, unfortunately, those women that died of the overnight abortion pill and creating that splash, and people now looking into it. And I think we're part of that, contributing this information. And now physicians are at least aware if a young woman comes in, she doesn't feel well, she has a high white cell count, they don't put her on antibiotics immediately because at least they're familiar with this pathogen. In fact, when I got to be part of this spotlight a little bit, and I got interviewed and we were put in a magazine or a journal describing what we thought was going on. At the same time, Planned Parenthood also had a portion in this journal. We were kind of talking back and forth about the subject. If you were to read the back of the overnight abortion pill, the RU486, it now describes Sordellii as a pathogen that's been associated with these infections and the symptoms, et cetera, because we have more information. But even talking to physicians now, some have heard of it, some aren't quite familiar with it. And so getting a high white cell count isn't just immediately associated with Sordellii, at least initially. By the end, I'll show you. Okay. I don't know how to go back. There we go. But initially, the whites, it doesn't go to 200 immediately. It kind of gradually starts to increase. And so these counts are around the peak of the infection when it's almost too late to save them. So as they're starting to climb initially, I think that could be one of several things. And so physicians aren't keen to just immediately assume that it's a Sordellii infection. I think once they start to get really high, then they know what's going on. But by that point, it's too late because you can treat bugs, but you can't treat the toxin. There's too much toxin circulating, and while you can give antibiotic to kill Sordellii, it's pretty resistant, or sensitive to most antibiotics. The antibiotics don't treat the exotoxins that are circulating. And so you go to these conferences again, there's a big talk is always developing an antitoxin for these difficile infections. If you can develop something that actually targets the toxin, you might be able to save these people. So again, we're working on that. If you can come up with that, I think you'd be a very rich person.
So is what the thing that's happening here is that this RU486, is it killing possibly something good, some micro, some
Sure, flora.
That's part of the flora that's keeping that Sordellii in line?
What we think, you guys know Warren Buffett. He funded a study. We got to be a part of that. We think that Sordellii is part of the natural flora for women, the vaginal flora. And so when women give natural childbirth, or in this case, undergo a medically induced abortion, it opens up the cervix and it creates an environment to where it's a niche for these floors to, again, kind of get in places where they shouldn't. And so that was what we believed. And so Warren Buffett funded this study where they cultured, I forget, it's like 20,000 women between California and Florida, the whole United States, to get a feel as to what women carried, how many women carried as part of the flora. Because I should also say at this time, something like 11 women, it's not like 200 women came down with these infections, something like 7 to 11 women came down with these medically induced associated Sordellii infections. But in Europe, they've been doing this type of stuff for almost a decade, and they don't have one record of a Sordellii-related infection. So we thought, well, it's an epidemiological thing. It must be more in the United States and in Europe. And in fact, 7 of the 11 women that died of these infections were from California. The 3 heroin outbreaks were all from California. So we thought, oh my gosh, it's probably in the dirt in California and nowhere else in the country. So we did this study, and they could hardly find Sordellii on, I think, one woman out of the 20,000 they found as part of their flora. So then that kind of reeled in that hypothesis, like, well, maybe it's not. So don't really know for sure, but we think it's part of the flora. But again, we're not too familiar, and then all the funding has really stopped to study this bug because it's so Okay, so you guys know the innate immune system. Again, that's kind of your initial line of protection, against invading pathogens. And so you need to mount that in order to fight things initially. Otherwise, they'll grow unchecked, and that's when you need the antibiotic in hopes of clearing these infections. But they're responsible, the innate immune system, for things like swelling, inflammation, fever, and pain. Again, often accompanies the innate immune response. And again, these patients don't seem to know that. Now the leukemoid reaction, as I quickly described, it's just an extremely high white cell count, greater than 50 times 10 to the third. It often resembles leukemia, but it's due to something more acute, like infection. Sorghum infections, huge white cell counts, and the majority are granulocytic, the white cells. I don't know if you're familiar with neutrophils versus monocytes, lymphocytes. Majority of them are the neutrophil or neutrophilic lineage. It's because that's mostly what's in circulation fighting infection for you, okay? And it was the only significant predictor of fatal outcome for these infections. I forgot to mention that. So if you're developing a high white cell count during this infection, chances are you're not going to make it, okay? So this is an example. We also put out the black line. Is a 23-year-old woman that gave natural childbirth. The white dots there, I think it was a 5-year-old boy that was playing on his dad's parked pickup, and he fell and he broke his arm in the dirt. They both developed Sordelii infections, and you can see the dynamics of the white cell count over days. This happened to be over days. But they both died on their respective last points of their white cell count because it became so aggressive they couldn't stop it. By the time they figured out it was actually happening, both died from their infections. And so you can see early on how it's kind of a lower white count, so kind of hard to tell, but then as it starts to really grow, I think they become comfortable physicians with what's going on, but it's typically too late.
Did you guys ever, sorry.
No, why?
Did you ever find anything why they weren't mounting sort of?
I just published this paper. Yeah, exactly. Yeah, good question. And so we're hoping, again, it's unfortunate. I mean, people are dying, and I totally respect that. But if there was more of a problem, I think we would be getting a lot more funding to further these studies, but it's just not happening. But what is it? I think there's $1 billion for Zika virus research now because there was one case in the United States, and I get it, but okay, anyway. So we were lucky enough, or we developed a murine model or mouse model of this infection. You can inoculate mice, which are amazing. It's funny, aside, using mouse models or animal models are great, very key for research, but it's amazing what a mouse can withstand. It takes 10 to the 6th bacteria to actually cause a fatal infection in a mouse, but it probably takes, let's say, 1,000 bugs to kill or just create a fatal infection in humans. So very different innate immune responses between mice and humans. And so all this data that we all collect as researchers, you have to take with a grain of salt because you can't, it's not just an automatic lateral transfer. Well, this happened in the mouse, so this is exactly what's happening in humans. Not the case, because they have a very different immune system, and much better, it seems, at least with these pathogens than we as humans have. But nonetheless, developed the animal model. We were creating fatal infections in these mice. They were developing these high white cell counts, again, not as robust as what we see in humans, but nonetheless, we're able to show increases in white cell counts, and they correlated with the timing of death. As they started to spike, these animals were crashing. So the same phenomenon we observed in human cases. And so we were trying to describe or figure out exactly what's causing this leukemoid reaction, what's going on. And so, oh, my, that's missing. Oh, there we go. I messed up that animation. Okay. So we try to explain it from 2 points of view. Was the bug producing something that actually causes white blood cells to proliferate, or is it something where white blood cells were escaping from the bone marrow in an unregulated type of manner? So that was all my PhD stuff. So this slide represents like 1 year's worth of work. But what we were able to show is that the neuraminidase produced by the Sordelii organism, when you expose promyelocytic cells, it's this HL60 cell line, which are still capable of proliferation, if you expose them to to this neuraminidase, you get this proliferative response greater than the control and equal to, this is our positive control, granulocyte macrophage colony stimulating factor, causes these guys to proliferate too. So we were getting an equal type of proliferative response, and if you put the two together, we had this huge proliferative response. So we thought, well, maybe this is what's causing this reaction. This I got all messed up. So this is the Clostridium mutagenesis. This is basically a system that we use to knock out genes in Clostridium. It's very, very useful. It was developed by a collaborator that we work with in the University of Nottingham in England. And Clostridium have been so difficult to create mutants in forever because of the thick cell wall, these gram-positive organisms. Hard to introduce things in there. But they've developed this system that has allowed us to do that. And so this is a slide basically to show that we were able to knock out the neuraminidase gene. And so when we did that, so let's start with this slide here. What happens, I'll start here. You get the wild type, you get the leukemoid reaction for the wild type infection. The neuraminidase mutant, you still see the leukemoid reaction, but it's a little earlier, and that's because these neuraminidase mutants are more lethal than the actual wild type. And so I haven't published this paper yet, but I think once I do, I'll get another phone call from somebody in Washington, because it's like the perfect, it's a highly lethal organism. And again, spore former, and there's no real signs or symptoms associated with initial infection. And I think that's dangerous to get a hold of. And I bet you that's going to cause conversations. Yes, ma'am.
This may be a dumb question, but is wild type mean?
I'm sorry.
Naturally, and then the other is what you create?
Exactly. If the wild type is the normal organism, the one that we initially started with, and then the delta stands for loss of, and then that's my mutant, which was created from the same kind of parent strain, if you will, the wild type strain. So,
I thought that was one that went to Mars.
wild type. That's the next lecture next week. So it clearly wasn't a proliferative response, and so we really now think and can almost show that it's due to an unregulated egress of white cells that are kept in the bone marrow. Okay, and so there's a highly specialized, very thin endothelial cell layer that separates the vascular system from the bone marrow, and any sort of damage to that will cause this watershed dumping of these white cells. And so we've shown that if you grow endothelial cells, in vitro in a petri dish, and you expose them to the toxins from Sordelii, you get this breakdown, as we already know. And so are comfortable now, or really our hypothesis is surrounding this idea of this dumping phenomenon. And again, these large clostridial cytotoxins, these lethal and hemorrhagic toxins that I talked about earlier, produced by Sordelii, are what cause these roundings. And notice that it's part of a small family of toxins called large clostridial cytotoxins. Only a handful of organisms produce it, including difficile. Okay, and that's what's allowed me to kind of transition from the Sordelii to the difficile research world, because of the similarity between these exotoxins, and because patients with difficile also develop high white cell counts.
Is there a way to check the levels of the white blood cells in the bone marrow? To measure and then measure it?
Before and after.
Right.
Yeah, so hopefully this grant gets funded, and that's what we'll be looking at. Part of that is to look at the actual bone marrow itself and use different markers to try to determine which populations are being shed and which ones are trying to repopulate.
Okay.
But I haven't done that yet. But that's right, to show that that's in fact what's happening. Question? Oh, okay. That was cool. So anyway, that's these toxins. And so what happens in the bone marrow too, you have these granulocytes, and they're tethered to what's called bone marrow stroma, these stromal cells, and they're responsible for kind of tethering these things to the bone marrow until, for reasons not really clear, it's very dynamic, and there's a lot of research to be done in that poetic world. Once the signaling is appropriate, these adhesins, or these receptors, this SDF1 and the VCAM1 produced by the stromal cell, and the CXCR4 and the VLA4 produced by the granulocyte. It's these relationships that keep it tethered. And once the signaling the timing is appropriate, this interaction no longer takes place, and you get natural release